Good morning. This is your final revision video for the final revision test, and I'll be focusing more on questions and answers. I'll be trying to minimize the editing to minimize the duration of the video as much as I can. So here we have on the slide the anatomy of the heart. And of course, one of the famous questions that you might encounter on the test is to describe the anatomical location of the heart. So number one, describe the anatomical location of the heart. So when I say location, I mean where exactly is the heart located? So first of all, we should say that the heart is located inside the chest cavity. And then we should locate the surfaces of the heart in relation to the surrounding structures, meaning that when you answer that question, which is to describe the anatomical location of the heart, you are at the same time describing the surfaces of the heart as well as the neighboring anatomical structures that surround the heart. So it's actually two big questions in one question. So after having said that the heart is located inside the chest cavity, you should say that the anterior surface of the heart lies behind the sternum, whereas the two lateral surfaces of the heart are related to the inner surfaces of the right lung and the left lung. The posterior surface of the heart, or the base of the heart, lies in front of the vertebral column and the esophagus. Actually, the esophagus first, and then more posteriorly, the vertebral column. And then the inferior surface of the heart lies on the diaphragm. So it is related to the diaphragm. And that's why we call the inferior surface of the heart the diaphragmatic surface of the heart. So how many surfaces have we mentioned now? We have mentioned number one, the anterior surface of the heart. This is number one. Number two, the two lateral surfaces of the heart. So up till now, we have three surfaces. The posterior surface or the base is the fourth surface. And then you have the diaphragmatic or inferior fifth surface of the heart. Here, of course, you are requested to draw the heart, but the drawing of the heart will be more related to the inside of the heart and to the flow of the heart. So if I ever ask you to draw the heart, I would rather ask you to draw the interior of the heart after having taken a longitudinal section of the heart, which is this diagram. So this diagram, which is the diagram that shows the flow, this is how the question will be designed if I ever ask you to draw a diagram representing the flow of blood inside the heart. And of course, when we describe the flow of the heart, the blood inside the heart, we have to mention the role of the atrioventricular valves, as well as the role of the semilunar valve, the semilunar valve. It's true that the question is about the flow of the heart inside the flow of the blood inside the heart, but the flow of the blood from the inside of the heart to the aorta and to the pulmonary arteries should also be mentioned, because if it is disturbed, it will affect the rest of the flow of blood inside the heart. Coming back to that diagram, which important question can be asked here? I might ask you to label the diagram. So of course, you should know that this is the surface anatomy of the heart. Here you have the right ventricle, and here you have the left ventricle. And in between them, you have a groove, an oblique groove, where you have this branch of the left coronary artery, and we call it the anterior descending branch, the left anterior descending branch of the coronary artery. The left anterior descending branch of the coronary artery lies in the groove, in the surface groove between the right ventricle and the left ventricle. And then here we have another horizontal groove that separates the right atrium from the left atrium. And in that groove between this transverse groove will separate both the right atrium and the left atrium above from the two ventricles below. So again, this horizontal groove that runs all around the heart separates the atria above from the ventricles below. So in the groove, in this part of the groove between the right atrium and the right ventricle, you have the right coronary arteries. And in the other part, you have an artery whose name is the left circumflex artery. You needn't know that name, but if you know it, of course, that would be great. All these are branches of the coronary arteries. As you might already know, that the coronary arteries are very small arteries that supply the myocardium of the heart with blood, and therefore they supply the myocardium of the heart with oxygen and with glucose to get the necessary energy to contract. So the coronary arteries are of extreme importance. Unfortunately, we didn't have time to study slides, I think from 102 to 104, where we were supposed to talk about the ischemia of the heart or the diminished blood flow to the heart. If it is relative ischemia, you needn't know this for the test. I'm just mentioning because it is important to know. If the ischemia is relative, not complete, then the patient will have chest pain. But that chest pain is reversible, and the condition is called angina pectoris. Angina pectoris. This is because the coronary arteries have fats deposited inside, and there is diminished surface area for passage of blood. This is an important medical condition, but more dangerous than this is when you have absolute ischemia, when no blood at all passes because there is complete obstruction of the coronary arteries, in which case we call this myocardial infarction, because infarction here means death, and this is because the myocardium of the heart or part of the myocardium dies because there is no arrival of blood at all to the myocardium. This is not included in the test, but I found it important to mention because we cannot finish the circulatory system without having mentioned two important diseases, angina pectoris and myocardial infarction. Myocardial infarction is an emergency condition in medicine. The patient should be hospitalized in an emergency or intensive care unit, and he should take medications to dissolve the blood clot that caused complete obstruction of the blood flow to the heart. Coming back to our diagram and the labeling of the diagram, you are supposed to you are supposed to know the blood vessels that are connected. So here, if this is the right atrium, definitely this is the superior vena cava. And if this is the right ventricle, so definitely this is one of the branches of the pulmonary arteries. And if this is the left ventricle, then this is definitely the aorta, which is, what is the function of the aorta? It is the largest artery in the body, and it delivers oxygen and nutrients to the whole body in the systemic circulation. So the blood in the aorta is what kind of blood? It is oxygenated blood. What is the function of the pulmonary artery? This is another sub-question from the diagram. The function of the pulmonary artery is to deliver deoxygenated blood to where? To the lungs, where there would be the alveolo-capillary exchange, so that the blood that returns to the heart via the pulmonary veins to the left atrium would be deoxygenated blood. And this is the content of another question that will be coming very soon, which is, how is the deoxygenated blood in the pulmonary artery converted into oxygenated blood in the pulmonary veins? Another important concept here, or side item here, is that the pulmonary artery is the only artery in adult persons that carries deoxygenated blood. How does oxygenated blood return to the heart? It returns via the pulmonary veins that we don't see here. And the pulmonary veins are the only veins that carry oxygenated blood. So again, the pulmonary arteries are the only arteries that carry deoxygenated blood, and they carry this blood to the lungs to unload it from carbon dioxide and load it with oxygen. And the pulmonary veins are the veins that they are called veins. Why? Because they carry blood from an organ to the heart, and they carry oxygenated blood. The superior vena cava delivers deoxygenated blood from the head and neck and the upper part of the chest to the right atrium. But there is another important vein on the same straight line coming from below, which is the inferior vena cava, that carries deoxygenated blood from the lower part of the body. So the right atrium actually is the site where all the deoxygenated blood from the body is collected to be sent to the right ventricle and become oxygenated. What are the branches of the aorta? The branches of the aorta, just in case you have to label. So we have here the left subclavian artery that passes under the clavicle to the left arm. And then we have here the left common carotid artery that goes to the brain. Both are left, by the way. And then we have here the brachiocephalic artery. This one is the brachiocephalic artery. And this brachiocephalic artery, the right brachiocephalic artery, will divide into the right common carotid artery, right common carotid artery, and the right subclavian artery. So there is a difference between the left side and the right side. On the left side, the left subclavian artery and the left common carotid artery arise from two separate points on the aortic arch. Whereas here on the right side, we have a common stem first, which is the brachiocephalic artery, from which we have the right common carotid artery and the right subclavian. So here you have left common carotid, here right common carotid. Again, left common carotid, right common carotid. Here you have the left subclavian artery, and here you have the right subclavian artery. But the only difference is that the left subclavian artery arises directly from the arch of the aorta, whereas the right subclavian artery and the right common carotid artery arise from a common trunk. Concerning the borders and the margins of the heart, we have talked about the coronary sulcus, the horizontal coronary sulcus that separates the atria from the ventricles. So this is the function of that coronary sulcus, and you have to know the arteries that circulate in it. And then you have the anterior interventricular sulcus, where you had the left anterior descending artery, and you have another one like this one at the back, which is the posterior interventricular sulcus. You can omit the margins and the borders because they are advanced anatomical definitions, and I would not ask you about them in an assessment. Concerning the inner heart anatomy, of course, it's important because I might ask you to describe. This is the second big question. Of course, we mentioned the first big question, which is the location of the heart and the anatomical relationships. But under this big question number one, there were sub-items here concerning the inner heart anatomy. You would be asked either to draw the inside of the heart or to describe the flow of the heart inside the flow of, I always say the flow of the heart, the flow of blood inside the heart, the flow of blood inside the heart. Or I might simply ask you to describe the inner heart anatomy. If I just ask you not to describe the flow of blood, it would be much easier. If I say describe the inner heart anatomy, you just describe the chambers, the two atria, the two ventricles, the interatrial septum, the interventricular septum, the valves, but you would not describe the physiology. So this is the difference between describe the flow of blood inside the heart. Here you are describing the function, you are describing the physiology. But if I say just describe the inner heart anatomy, then you are describing the structures. Now, let's answer first, describe the inner heart anatomy, which is the easier description. So inside the heart, we have four chambers. We have two upper chambers, which are the left atrium and the right atrium. And we have two lower chambers, which are the right ventricle and the left ventricle. In between the two atria, we have a separating muscular wall, which is the interatrial septum. And in between the two ventricles, we have another muscular wall, which is the interventricular septum. Furthermore, between each atrium and its corresponding ventricle, we have an atrioventricular valve. So we're going to talk about the atrioventricular valves. And then you would say that on the right side, we have the tricuspid valve, which is made of three flaps or three cusps, whereas on the right side, we have the mitral valve or the bicuspid valve, which is made up of two flaps only. Have we finished the internal structure of the heart? No, not yet. We still have to mention the valves between the ventricles and the large arteries, because we have just talked only about the atrioventricular valve. So in order to finish, we have talked about the upper chambers, we have talked about the lower chambers, second point. Third point, we have talked about the interatrial septum, and then fourth point about the interventricular septum, fifth point about the atrioventricular valves, and mentioning their names. But we wouldn't mention the function if we're asked only to mention the structure. And then last thing, we would talk about the semilunar valves. What are the semilunar valves? They are one-way doors allowing the flow of blood in one direction only. But you are not required to mention this if you are only asked about the structure of anatomy of the inside of the heart. And they are called semilunar valves because they are made of cusps that in the cross section look semilunar. And we have the aortic valve between the left ventricle and the aorta, and we have the pulmonary valve between the right ventricle and the pulmonary artery. Having mentioned this, you would have finished the inside anatomy of the heart. But if you are asked about the flow of the blood or the physiology inside the heart, unfortunately, you should start by mentioning the anatomy. You cannot start immediately by saying deoxygenated blood passes from the right atrium to the right ventricle, and oxygenated blood passes from the left atrium to the left ventricle. You should mention the anatomy first. So of course, when you're required to mention the flow of blood inside the heart, unfortunately, you have to mention the anatomy first. Now, let's answer the question of describing the flow of blood inside the heart. First thing, you have to mention the importance of the septa. Both the interatrial septum, and we have two septa. We have a septum between the two atria, and we have another septum between the two ventricles. So you should start by mentioning the importance of the septa. They prevent mixing of deoxygenated blood on the right side of the heart from oxygenated blood on the left side of the heart. So this is important to know. And then describing the flow, you have to start at one point because it's a cycle. So you have to choose a point to start at. I would rather start by or at the superior vena cava and the inferior vena cava. I would start here and say that deoxygenated blood comes through the superior vena cava and the inferior vena cava to the right atrium. And then deoxygenated blood passes across an open tricuspid valve to the right ventricle. And then the right ventricle pumps this deoxygenated blood through an open pulmonary valve to the pulmonary artery. But at that time, the tricuspid valve should be closed. So see how important it is to go step by step. So when the right atrium pumped deoxygenated blood into the right ventricle, there was an open tricuspid valve. And then when the right ventricle pumped the received blood during systole into the pulmonary artery, it was through an open pulmonary valve. But meantime, you should mention that there was a closed tricuspid valve. And then blood is unloaded from oxygen and unloaded from carbon dioxide. So in the lungs, there is unloading from carbon dioxide, and there is loading with oxygen, so that the blood that returns to the left atrium via the pulmonary veins becomes oxygenated blood. Why have I mentioned it that rapidly? Because it's part of the total flow inside the blood. But if you're ever asked about the alveolo-capillary exchange in the lungs, it would not be enough to say, just to say unloading from carbon dioxide and loading with oxygen. You should mention diffusion, down concentration gradient, and we will be doing this. Now the oxygenated blood returns to the heart via the pulmonary veins. It's collected in the left atrium, and then it passes via an open mitral valve into the left ventricle, and then the left ventricle during systole will pump the blood via an open aortic valve into the aorta to be distributed to the whole body. But meanwhile, there is a closed mitral valve. So this is how you should describe the flow inside the heart. And now we're going to talk about something very important, which is the normal heart X-ray. Of course, we study why do we study a normal heart X-ray? Because we want to know the abnormalities. If you don't know the normal picture of the heart X-ray, you wouldn't detect any abnormality. But there are precautions to take, and these precautions that are mentioned by the technician to the patient are very important in order to avoid incorrect diagnosis or in order to avoid misdiagnosis. And one of the most famous incorrect diagnosis is cardiomegaly or enlargement of the heart. I mean that it would be falsely, of course, it might, the heart might be truly enlarged, but I'm talking here about the condition when you don't take the necessary precautions and the patient is not in deep inspiration and the diaphragm is not downward enough, so the heart appears larger than normal. So there are precautions that we should take to standardize the X-ray picture of the heart so that when you measure your measurements of the cardiothoracic ratio, they reflect a correct diagnosis and a correct size of the heart. So first of all, what are the precautions? First of all, the precautions are so and so, but they are important. Why? To avoid misdiagnosis, for example, to avoid an incorrect diagnosis of false enlargement of the heart or cardiomegaly. The first precaution is that the patient should be in deep inspiration, in the deepest inspiration, the end of inspiration, when the patient takes in his breath and cannot go any further downward. And then it's not enough to say the end of inspiration, but the patient would hold his breath afterward, okay? The second important precaution is that the clavicles should appear on the X-ray symmetrically. Why? First of all, how can we detect this? So if this is the manubrium sterni and this is the left clavicle and this is the right clavicle, so in the X-ray, they should be equal in length. And the manubrium sterni should be central. If they're not equal in length, if the left clavicle appears shorter than the right clavicle, then the patient is turning towards the left more. And that's why you don't see enough length of the left clavicle. And the reverse is true. So the importance of having equal length of the clavicles is to ensure that the patient is centralized. Because if the patient is not centralized, then the whole mediastinum is not centralized. Then the heart will be rotated towards the left or towards the right, and the whole mediastinum, the heart, the esophagus, the thymus. But what is important for us now is the heart. So, sorry. If the heart is rotated, you cannot estimate the cardiothoracic ratio correctly. How can you know as a medical doctor, as a radiologist who is inspecting the X-ray and examining it, that the patient was centralized when the X-ray was done by the technician? Just look at the level of the diaphragm. So this paragraph is the answer of how can you know, how can you confirm centralization of the patient? How can you confirm centralization of the patient? I can confirm centralization of the patient by checking the level of the diaphragm and the symmetry of the clavicles. So again, we're going to talk about the symmetry of the clavicles. If the diaphragm is at the level of the 8th to 10th rib posteriorly, then the patient is in deep inspiration. Or, in other words, if the diaphragm is at the level of the 5th to 6th anterior rib. Which one of these two levels is easier to detect? Posteriorly. The posterior ribs are easier to detect in the X-ray than the anterior parts of the ribs. There is nothing like a posterior rib, but I mean the posterior part of the rib. Here you have the 12 pairs of ribs. Of course, you know that we have 12 pairs of ribs. This is the clavicle. So this is important to detect because if you're counting the ribs, for example, to detect the intercostal spaces, you wouldn't count the clavicle. And don't forget that the first rib is just under the clavicle. And don't forget that the space between the first rib and the second rib is the first intercostal space. And therefore, the second intercostal space would be the space between the second rib and the third rib. The first intercostal space is between the first and second ribs, and the second intercostal space is between the second and third ribs. And then don't forget that 1, 2, 3, 4, 5, 6, 7 ribs. The upper 7 ribs are directly connected to the body of the sternum. This is the body of the sternum. This is the manubrium sterni, and this is the sternal angle, or the angle of Lewis. Why is the angle of Lewis important? Because there is a ridge here, like a bump here, and that bump, if you go downward with your index finger and find the bump, and then descend a little bit to the right or a little bit to the left, you would find the second right intercostal space and the second left intercostal space. Why are they important? They are important, as you know, in auscultation, because the second intercostal space is the space where the second right intercostal space, you have to mention the site, is the site where you can best hear murmurs from the aortic valve, and that's why we call it the aortic area. Similarly, the second intercostal space to the left is where you can best hear the pulmonary sounds. Now, if we look at the X-ray, we will find that here, for example, you will find that the posterior ribs are better visualized. They're better visualized. Why? Because, of course, they're better visualized because they appear more radio-opaque. Don't say because they are more radio-opaque, because all the bones have the same density and therefore are similarly or equally radio-opaque, but they look more radio-opaque because don't forget that you take a postero-anterior view of the chest, meaning that the device is put behind the chest. Of the X-rays are coming from behind, so those posterior ribs are better visualized, and therefore, and they are better. They are more or less horizontal. So another question is to justify why is it easier to visualize the posterior part of the ribs, and therefore it is easier to count from posterior because they appear better, they appear more radio-opaque. How can I know the posterior part of the ribs? By being more horizontal. On the other hand, the anterior part of the rib, as you can see, is more downwards and oblique like this, and it appears less radio-opaque, and therefore you cannot count them easily. If I get that X-ray and ask you whether the patient is in good inspiration or not, you would start by the clavicle. Under the clavicle, we have the first rib. This is the horizontal part of the second rib. This is the horizontal part of the third, horizontal part of the fourth, fifth, sixth, seventh, and eighth rib. So the level of the diagram is at the level of the eighth rib, which is okay. At that time, and is the patient centralized? The second precaution, yes, the patient is centralized, because if you measure the length of the clavicle here, it's equal to the length of the clavicle there. What's the importance of the centralization of the patient? The importance is that the heart is not rotated. And now we can estimate whether the heart is enlarged or not. Doing what? How? This is another question. How can you know or estimate whether the size of the heart is normal or abnormal or enlarged? By measuring the cardiothoracic ratio. This is important to know. What is the cardiothoracic ratio? The cardiothoracic ratio equals what? Equals the largest transverse diameter of the heart, the largest transverse diameter of the heart divided by the largest transverse diameter of the thoracic cavity. Concerning the thoracic cavity, it's easy to measure. This is a measurement. This is not a conclusion. This is not a logical sequence. So the largest diameter of the heart is easy to estimate. You just find the two most outbulging points and you take a transverse line between them. Say, for example, it is 10 centimeters on your X-ray. But the problem is with the largest transverse diameter of the heart, because the most bulging points of the heart on the right side and on the left, to the right and to the left, are not on the same level. Mostly you have the bulging point like this here on the right side, and the most bulging point is towards the apex there. So what are we going to do? We are going to draw a vertical horizontal line, a vertical line that is perpendicular to the diaphragm. And then we are going to take this transverse hemi diameter of the heart and another hemi diameter of the heart. Say, for example, this is A and this is B, and we're going to measure them with our ruler. So say, for example, that this one on the right side was 4.2, for example, and the other one to the left side was 5.6. So A plus B equal 9.8. So your cardiothoracic ratio would be 9.8, which is the largest transverse diameter of the heart, divided by 10, which is the largest sorry, not 9.8, this is too large. Sorry. We want it, if it's normal, we want it to be less than 10. So if the largest transverse diameter of the heart is 10, so say, for example, that this would be 2.4 and this would be 3.2. So when you sum up A and B, you would find 2 plus 4 plus 3.2, which is 5.6. 5.6 is the transverse diameter of the heart. 10 is the largest transverse diameter of the chest cavity. So in this case, the cardiothoracic ratio would be 5.6 divided by 10. This is larger than 50%, and therefore the heart is slightly enlarged. If it were, for example, 4.9, it would be less than 5, and therefore the heart would be of normal size. So when you have to calculate or measure the cardiothoracic ratio and calculate it, you have to write the rule, please. So the law is the largest diameter of the heart divided by the largest transverse diameter of the chest cavity. I'm not sure whether we covered mitralization of the left border of the heart, but in a normal chest X-ray, you should have like here, like an inside notch where the left atrium is present. If it is straight forward like this, if you have a straight line, so we say that the left border of the heart is straightened, and this happens when the left atrium is enlarged. And one of the commonest causes of left atrial enlargement is, if this is the left atrium, is a narrow mitral valve. So blood accumulates in the left atrium. So mitral stenosis is one of the commonest causes of enlargement of the left atrium. And therefore, mitral stenosis is one of the commonest causes of straightening of the left border of the heart, or that's why it's called mitralization of the heart. Now, this slide on the pericardium leads us to the structure function adaptation of the heart. So one of the questions that are frequently encountered on the test is the question on the structure, function, adaptation of the heart. So the first, you have like 6 or 7 or more points to mention about the structure, function, adaptation of the heart. And by this, we mean how the structure of the heart is well adapted to carry out the function of the heart. First of all, we start from outside and we talk about the tough membrane that lies around the pericardium. So first of all, we have a tough sac-like membrane around the heart. I'm talking now focusing on the structure, function, adaptation of the pericardium, which is part of the structure, function, adaptation of the heart as a whole. So concerning the pericardium, it is tough, and therefore it is a layer that protects the heart against trauma. Second, this membrane is a double membrane, and being a double membrane allows that membrane to secrete and keep fluid in between the two layers. So if this is the heart, there is a membrane that is adherent to the heart, and that is called the visceral layer, just like we did in the pleura. And there is an outer membrane that is called the parietal layer of the pericardium. And in between the two layers, we have fluid. So the fluid is secreted by the pericardium, but the presence of a double membrane helps this fluid to be kept in between the membranes. And it is important because the heart is all the time beating. So this fluid minimizes the friction between the two layers of the heart. And then we will pursue our discussion about the structure, function, adaptation of the heart. And in order for you to make your life easier, you can just do it in a logical way, and it needn't be memorized. How would we do this? If we draw the simplest diagram of the heart like this, and we draw the right atrium, for example, and then the left atrium, and the interatrial septum in between them, and then we draw the right ventricle that takes most of the anterior surface of the heart, and that has a certain thickness of the myocardium. And then we draw the left ventricle, and there is a smaller portion of the cavity of the left ventricle that appears. And notice that here we have a greater, so this is the cavity of the left ventricle. So if we go step by step, so the first structure, adaptation of the heart, and the simplest one, is that the heart is made of muscles. How is this important for the heart? Muscles allow the heart to contract because muscles contract and relax. So when the muscles contract, this allows pumping of blood from one area of the body to another. And when these muscles relax, they allow filling with blood. This is number one. Number two, the muscles are branched. The muscles of the heart are branched. And at the same time, they are interconnected. And branching serves interconnection. If they weren't branched, it would be much more difficult for them to interconnect. So if you see them under the microscope, you will see them as striated muscles, and they are branched like this, and they are interconnected with the neighboring muscles. This interconnection allows what? It allows the rapid transfer of ions, and therefore the rapid transfer of electric impulses from one muscle to another, and this helps the whole heart contract as one unit. And contracting as one unit, for example, when the two ventricles contract simultaneously at the same time, this increases the efficiency of the heart. The third thing that is related to the muscles is that the left ventricular wall is much thicker, and this is a common question to justify why the left ventricular wall is thicker than that of the right ventricular wall. When you have a thicker musculature, then you can pump more strongly. So this results in stronger contractions, and a stronger contraction is important because the left ventricle is facing two challenges. It is facing the challenge of the pressure and the challenge of the distance. The challenge of the pressure and distance are both present in the systemic circulation, which is the major circulation that spreads or distributes blood all over the body. The pressure is higher in the systemic circulation, being 120 millimeters mercury maximum systolic pressure obtained during systole and 80 during diastole. This is much higher than the pressure in the pulmonary circulation. The distance is another challenge. I have to have a thicker musculature because I'm pumping blood over the whole body, so I'm pumping blood across a greater distance. So this was the third item in the structure, function, adaptation of the heart, but we still have other items, the presence of the interatrial septum between the right atrium and the left atrium, as well as the presence of the interventricular septum between the right ventricle and the left ventricle are very important because they prevent mixing of oxygenated blood and deoxygenated blood. Furthermore, number 5, the presence of the one-way valves between the atria and the ventricle. The atrioventricular valves allow one-way passage of blood from the right atrium to the right ventricle during diastole and prevent backflow during systole. But you also have the number 6, you have the semi-lunar valves. They also regulate the flow of blood from the ventricles to the corresponding large arteries. So we have the pulmonary valve, which allows passage of deoxygenated blood from the right ventricle to the pulmonary artery during systole, and you have to say during what, and prevents regurg during diastole. Similarly, we have the aortic valve that allows the passage of the oxygenated blood from the right ventricle to the aorta during systole and prevent regurgitation or backflow during diastole. And this is again how you should describe the function of a valve. And then number 7, we have this narrow apex that is mostly made by the left ventricle and that allows maximization of ejection of blood into the aorta. You might add, for example, that the base of the heart or the posterior surface of the heart is strongly supported by the esophagus and by the vertebral column to keep the shape of the heart, whereas the right border and the left border, or the right surface and the left pulmonary surfaces, are supported by the right lung and the left lung to keep the shape of the heart. Having finished this, we have to mention one important question that is related to the interatrial and interventricular septum. So what happens when you have an atrial septal defect, ASD, or a ventricular septal defect? D stands for defect, and S stands for septal, and A and V stand for atrial septal defect or ventricular septal defect. So imagine that you have a normal interventricular septum, but you have here a defect in the interatrial septum. You have like an incompletely closed part of the interatrial septum. So in such a case, there would be mixing of oxygenated blood and deoxygenated blood. Since the pressure on the left side of the heart is higher than the pressure on the right side of the heart, so blood oxygenated blood would pass from the left atrium to the right atrium. So this is the first result, that oxygenated blood would pass to the left. This means what? This means that automatically less oxygenated blood would go, oxygenated blood would go to the right atrium. And then number two, you will have less oxygenated blood that passes to the left ventricle. And therefore, you would have less oxygenated blood into the aorta. This means that there will be less energy that reaches the body, and therefore the patient is all the time suffering from fatigue. The same occurs if you have a defect in the interventricular septum, but it would be, the symptoms would be more serious. But it's exactly the same description of consequences. We have just talked about the heart valves, and again, I emphasize that we have two atrioventricular valves and two semilunar valves. If you were to describe the function of atrioventricular valves in general, you would say that the atrioventricular valves allow the passage, in general, they allow the passage of blood from the two atria to the two ventricles during diastole. This is very important, during relaxation of the heart, and they prevent backflow that might occur during systole. So during diastole, the blood is allowed to pass. But during systole, when the ventricles start to contract, they should not allow the backflow of blood. They should close. So if I ever ask you, justify, it might be another type of question. It might be to justify the atrioventricular valves are open during diastole in order to allow passage of blood from the atria to the ventricles. Justify the atrioventricular valves are closed during systole to prevent backflow from the ventricles to the atria. But if I ask you to describe the function of the mitral valve specifically, or the tricuspid valve specifically, it's essentially the same frame, meaning that it allows during diastole and prevents during systole. But the only differences is that you have, number one, to mention the type of blood, whether it is oxygenated or deoxygenated, and the second point, the second specific point, that you have to say from the right atrium to the right ventricle, or from the left atrium to the left ventricle. So the specific items you add when you're asked about a specific valve is the type of blood, oxygenated or non-oxygenated, and the chambers. For example, if I ask you to describe the function of the mitral valve, you would say that it allows the passage of oxygenated blood from the mitral valve. I'm talking about the mitral valve. It allows the passage of oxygenated blood from the left atrium to the left ventricle during systole and prevents backflow during diastole. But if I ask you about the semilunar valves in general, so you would say that they allow passage of blood from the ventricles to the corresponding large arteries during systole. Of course, here it's the reverse. And so it's allowed during systole, and the backflow is prevented during diastole. But if I ask you about a specific valve in particular, like the pulmonary valve, you would add again the type of blood, oxygenated or deoxygenated, and the chambers. So you would say, for example, the pulmonary valve allows the passage of deoxygenated blood from the right ventricle to the corresponding artery, which is the pulmonary artery during systole, and prevents backflow during diastole. Similarly, if I ask you about the aortic valve, you would say it allows passage of oxygenated blood from the left ventricle to the aorta during systole and prevents backflow during diastole. So it's like 5 points that you have to mention specifically about each valve. Of course, the hemodynamics of the valves are very important. So we come back here, which is what we call the valve coordination. So I might ask you to justify the fact that the work of the valves related to the heart is coordinated. What do we mean by coordinated? We mean that they arrange together to make the flow of the heart in the good direction to ensure forward flow of blood. And when we say forward flow of blood, we mean in the good direction, in the correct direction. So one example of coordination is when the atrioventricular valves are open during diastole, during ventricular relaxation, but the semi-lunar valves are closed at that time. Or the reverse, you might say, atrioventricular valves closed during systole, and while the semi-lunar valves are open. So the plus sign here means meanwhile. And this is what we mean by coordination. They coordinate together to ensure smooth forward flow of blood. When the atrioventricular valves are open during diastole to allow passage of blood from the atria to the ventricles, by the way, on both sides. On the right side, the deoxygenated blood passes, and meanwhile, on the left side, the oxygenated blood passes. When the blood flows from the atria to the ventricles during diastole, the atrioventricular valves are open, but meanwhile, the semi-lunar valves, because of the coordination deal between them, should be closed. Because if they are not closed, they will allow backflow and there would be turbulent flow inside the ventricles. Of course, when we come to the alveolo-capillary exchange, and it's an easy concept that we have mentioned several times, but I needed to be mentioned in a particular organized way. So when you talk about alveolo-capillary exchange, always remember that diagram. We have the alveolus, okay, with the air coming inside and going outside, inhalation and exhalation. But overall, of course, the oxygen here in the air sac, in the alveolus, we are talking about the alveolo-capillary exchange. So you should have in your diagram an alveolus and a blood capillary. Suppose that the blood is coming from one branch of the pulmonary artery, so it is deoxygenated blood that is rich in carbon dioxide and poor in oxygen. And the blood is leaving on the other side. So this is the side of the pulmonary artery, and this is the side of the pulmonary veins when the capillaries will collect to form the pulmonary veins that go to the left atrium. And here the pulmonary artery takes deoxygenated blood from the right ventricle. So at this side of the pulmonary artery, oxygen is very low. So oxygen will diffuse from where it is in a higher concentration to where it is in a low concentration, down concentration gradient. So gradually, the amount of oxygen increases at the other side of the capillary. Meanwhile, we had a small amount of carbon dioxide here, but we had a large amount of carbon dioxide at the beginning. So carbon dioxide will diffuse the reverse way from the side where it is in a higher concentration to the side where it is in a lower concentration. So gradually, as we move forward after having done the exchange, the amount of carbon dioxide diminishes. So at the end of the capillaries where they are going to join with the pulmonary veins, you have a high concentration of oxygen because you have loaded your capillary with oxygen via diffusion down concentration gradient, and you have a low concentration of carbon dioxide because you have unloaded your capillaries from carbon dioxide. Now for the control of the heartbeats, of course, we're going to talk about the sinoatrial node. Of course, the heart beats in a cyclical way about 60 to 90 beats per minute. What controls the heart rate is the sinoatrial node. So first of all, why is the sinoatrial node considered the pacemaker of the heart? So first of all, you have to define the pacemaker of the heart. The pacemaker of the heart is the part of the heart that controls the heart rate. And the whole myocardium will follow the same rate as the pacemaker. So it controls, the word pacemaker means that it decides the heart rate. It controls the heart rate. It decides it first and then it controls it. And the whole heart follows the rate of the pacemaker of the heart. What part of the heart is the pacemaker? The sinoatrial node. The sinoatrial node can be described as a specialized cardiac tissue. It is a specialized cardiac muscular tissue, specialized cardiac muscle. Why is it specialized? Because it can generate impulses on its own without electric innervation. Not only this, but it can generate impulses at the fastest rate. So in this slide, we have defined the pacemaker and we have justified why the SA node is considered the pacemaker, because it is special. It doesn't have the same properties as the rest of the muscles. Special in what? In the fact that it can generate impulses spontaneously on its own. Not only this, but at the fastest rate, so that any other part of the myocardium that tries to be the pacemaker will fail to do so if the SA node is healthy. Okay, so the SA node is the pacemaker of the heart. How does it do this? It generates electric impulses on its own, and then these electric impulses will spread first over the myocardium. After having spread over the myocardium, the muscles of the atria contract. So this is another important piece of information. In order for the muscles of the atria to contract, they should first receive an electric impulse. And then the electric impulses are transferred with delay via the atrioventricular node to the two atria at the same time. And when to the two ventricles, at the same time. And when they receive their electric impulses, they contract. So this is how the SA node distributes the electric impulses on the myocardium. Or in other words, the question is, how is electricity spread over the myocardium? So this second part of the explanation on the slide is answering the question of the spread of the electric impulses over the myocardium. And here I have to mention two important things. The first thing is the atrioventricular node. The atrioventricular node is another specialized cardiac tissue, meaning that it also can generate electric impulses on its own. But the problem is, it's a problem and an advantage at the same time. It has a slower rate than the sinoatrial node, which ranges between 40 and 60 beats per minute. Why is it a disadvantage? It is a disadvantage for the AV node because it does not allow the AV node to be the pacemaker. Under normal conditions, it can't be the pacemaker of the heart under normal conditions. It's not a problem for the heart. It's not a problem for the human being. It's a problem for the atrioventricular node. But it's an advantage because this lower rate allows what? It allows many, many benefits. The first benefit is that it allows time for the ventricles to fill with blood, and this is important for efficient contraction. If they don't fill with blood, they wouldn't contract efficiently. And number two, it prevents, so it allows and prevents. It allows time for the ventricles to fill with blood, and it prevents simultaneous contractions of atria and ventricles. Of course, if there is simultaneous contraction of atria and ventricles, it would be a great problem because there would be turbulent flow and there would be inefficient pumping of blood by the ventricles. So this, and there is another third benefit that we haven't mentioned before, but of course you can deduce it now that you have studied the arrhythmias. Number 3, it protects, of course, the ventricles against very high rates of the atria. And we have seen this, for example, in atrial, against high atrial rates. This is the third benefit we haven't mentioned before. If you have atrial fibrillation or atrial flutter, and a very high rate here by an ectopic MAD focus in the atria that beats at 300 beats per minute, of course, the slow rate in the atrioventricular node is protective. But the fact that it is a specialized cardiac tissue is important because even though under normal conditions, it can be the pacemaker of the heart because of that slow rate, but if the sinoatrial node is diseased, for example, because of ischemia, but the AV node is still working well, then the AV node will take over and the AV node will be the new pacemaker of the heart, but it will drive the whole heart at a very slow rate. There remains one point, which is why do these structures, or why are these structures able to generate spontaneous electric impulses? Why are they specialized? They generate spontaneously, they generate the electric impulses spontaneously. Why? The big question is why? This is because they have an unstable resting membrane potential. And here we come to many definitions that are very important. So they have unstable resting membrane potential. This is an answer to what? This is an answer to why can the SA node and the AV node generate electric impulses spontaneously? Because they have an unstable membrane potential. And here we're going to discuss several items like the resting membrane potential, depolarization, repolarization. So what is a resting membrane potential? Under normal conditions, when the cell membrane, for example, the cell membrane of the myocardial muscle is resting, means that it does not receive stimulation. Resting means that it does not receive stimulation. And that's why we call it resting membrane potential. So when can we see or measure a resting membrane potential? We can measure it when the cell is resting, when the cell does not receive any external stimulation. So under normal conditions, the external surface of the membrane that is in contact with the extracellular fluid is positive in relation to the inside. And there is a potential difference of minus 70 millivolt. And the minus sign refers to the fact that the inside is negative to the outside. What does this mean? This means that does this mean that there are only positive ions outside the membrane? No. But this means that the overall algebraic sum of the charge outside is positive and the overall algebraic sum inside is negative. So there is a potential difference, a difference in electric charge between the outside and the inside that is negative inside, and the value is minus 70 millivolts. This is the resting membrane potential. But when the cell receives electric stimulation, what happens is that there is depolarization or reversal of polarity. So if there is an external, I'm talking about normal cells, I'm not talking about unstable cells. Under normal conditions, if there is an external stimulation, what happens is that there is opening of very rapid sodium channels that allow rapid entry of sodium ions from the outside to the inside if there is external stimulation. So what happens at the end of the day is that instead of having the outside positive and the inside negative, we will have the reverse. We will have the inside positive because of the entry of many positive ions inside, and the outside negative. This is the reverse of what we saw during the resting state, and this is why we call this status reversal of polarity. Reversal of polarity, or we call it depolarization. So we call it reversal of polarity. So what is depolarization? Depolarization is the status of the cell membrane when the inside of the membrane is negative to the outside. In such a case, for example, the potential difference would be plus 40 millivolt. And the positive sign here is an indication to the inside. So the sign before the value of the potential difference always indicates the status of the inside of the membrane. But this status cannot stay for long, because when you have a lot of positive charges inside, they start repelling with each other. And what happens afterward is that we return to the resting state, which is the last state, which is repolarization. In repolarization, what happens is that sodium channels, yes, close, and potassium channels open. Potassium channels open. And when potassium channels open, potassium finds a way to the outside because inside there are a lot of positive charges. And when potassium goes to the outside, we return to our original status, which is positive outside and negative inside, and that's why we call it repolarization. So repolarization means returning to the resting electric potential, which is outside positive, inside negative. But we still have a problem, is that we corrected the electric charge. We made it positive outside and negative inside, but we have a reversal of concentration, because now potassium, which is supposed to be the main cation inside or intracellularly, is outside. And sodium, which is supposed to be the main cation outside, is now imprisoned inside. And here comes the role of the sodium-potassium pump. So the sodium-potassium pump works after repolarization to pump the potassium ions again inside and pump the sodium ions again outside, so that you adjust the chemistry or the chemical gradient after having adjusted the electric gradient. So, sorry. The function of the sodium-potassium pump. So as you know, the sodium-potassium pump works after repolarization here. And it does what? It pumps 3 sodium ions out, in exchange for 2 potassium ions in. So it's a pump, it requires energy. It's an active process that is derived from the hydrolysis of ATP. And here we are returning sodium to its home to be the main cation extracellularly, and returning potassium to its home to be the main cation intracellularly. And therefore the sodium-potassium pump adjusts the chemical gradient after the electric gradient has been adjusted by opening of the potassium channel. Of course, the trigger was the arrival of an external electric impulse. What is specific about the sodium, what is specific about the SA node and specific about the AV node is that all this can occur spontaneously without arrival of an electric impulse. And that's why we say that their membrane is unstable, because the sodium channels open spontaneously without any external stimulation. Of course, we have covered all the previous slide that I run now, and systole is contraction and diastole is relaxation. Of course, both the atria and the ventricles contract, but when we say systole in general, absolutely, we are talking about ventricular systole. And when we say diastole, absolutely, we are talking about ventricular relaxation. And of course, we talked about the status of the valves during systole and during diastole. And now we come to the heart sounds. The heart sounds, we have two heart sounds. We have the first heart sound and the second heart sound. And we have two heart sounds. You have the first heart sound and the second heart sound. And the first heart sound is due to closure of the atrioventricular valves, and the second heart sound is due to closure of the semi-lunar valves. So we have lub-dub, lub-dub. Lub is the first heart sound, and dub is the second heart sound. And the first heart sound is due to closure of the atrioventricular valves. The second heart sound is due to closure of the semi-lunar valves. But you have to say when. The closure of the atrioventricular valves occurs at the start of systole, and the closure of the semi-lunar valves occurs at the start of diastole. Of course, as you know, if we draw an audiogram of the first heart sound and the second heart sound, and then the next cycle, S1 and S2, you will find that we said that the first heart sound is closure of the atrioventricular valves at the beginning of systole. So if this is the beginning of systole, then this is the whole duration of systole. So the distance between S1 and S2, what does this represent? It represents systole. And then S2, which is closure of the semi-lunar valves at the beginning of diastole. So this means that here we have the beginning of diastole, and that all this is the duration of systole. So what does this duration represent? It represents systole. And what does this duration represent? It represents diastole. And needless to say that diastole is longer than systole. This is normal because diastole is the time of relaxation of the ventricle. So if I ever ask you, why is diastole longer than this systole? First of all, it allows relaxation of the ventricles to prevent fatigue of the ventricles, and furthermore, it allows filling of the ventricles. So diastole is important for the ventricles to relax in order not to get fatigued, but at the same time to have enough time to fill with blood. How is diastole? This is an important piece of information. How is diastole affected when the heart rate is increased? When the heart rate is increased, both systole and diastole are shortened. Both are shortened because we have a bigger number of cardiac cycles per minute. But they are shortened at the expense of diastole. Meaning what? Meaning that diastole is more affected than systole. And this is serious. And this is why rapid heart rates are dangerous, because it's true that they affect both systole and diastole, but they affect diastole, the time of relaxation, more than systole, and therefore there isn't enough time for the ventricles to relax nor to fill with blood, and this results in ventricular fatigue and in diminished volume of blood that is sent to the body. Of course, this is a normal audiogram because you don't have any superimposed sounds on S1 and S2. But if you have a superimposed sound here or a superimposed sound there, then you have a murmur. What is a murmur? A murmur is This is important to define a murmur. It is an abnormal sound upon auscultation that is superimposed on S1 and S2, meaning that you listen to S1 and S2, and in between them, you have a murmur. Of course, murmurs are due to turbulent blood flow, non-laminar, non-smooth blood flow, and mainly due to valvular diseases. If you have a murmur between S1 and S2, then it is a systolic murmur. But if you have a murmur between S2 and S1, then you will have a diastolic murmur. The auscultation areas are very important. Angle of Lewis is very important to determine the second intercostal space to the right. This is the aortic area, or where you can best hear the sounds of the aortic valve. Second, right intercostal space, you have to say right, at the sternal border. Pulmonary area, second left intercostal space at the sternal border. Erb's point, third intercostal space at the left sternal border. So every time you have to say which space, right or left, and which sternal border. And then you have the apex, which is the mitral area where you can best hear the mitral valve sounds. And it is the fifth intercostal space, mid-clavicular line to the left. And then you have the tricuspid area, again, fifth intercostal space, but more to the inside towards the left sternal border. So describing the auscultation areas is very important, and drawing the auscultation areas is very important. So this is an additional diagram that you have to draw. The definition of the cardiac cycle is very important. It's a regular sequence because it is repeated. And during that regular sequence that is repeated, you have contractions and relaxations. And the contractions and relaxations are alternating, of course, because when the muscle is contracting, it cannot relax at the same time. And it is important to note that both the atria and ventricles contract and relax. They do not. Atria and ventricles do not contract at the same time, but the two atria, this is important to notice, the two atria contract at the same time, and the two ventricles contract at the same time. But between the atria and the ventricles, there is the AV nodal delay, and the final purpose is to pump the blood. And it starts, of course, at S1 and finishes at the next S1. So it's a sequence of sequence is important, regular is important, alternating is important, contractions and relaxations is important of the atria and ventricles to pump the blood. And it starts at the beginning of one heartbeat, like the beginning of an S1, for example, and it ends at the next S1. I promised you that I wouldn't ask you about this or what happens during the phases, but there are items that we have already covered, like, for example, when does the blood pass from the right atrium to the right ventricle? It passes during atrial diastole and during ventricular diastole at the beginning. And then the ventricle continues to relax while the atria begin contraction. If I ever ask you any question about the phases of the cardiac cycle, it would be one of the concepts we have covered before. For example, when does the atrium send to the ventricle? Of course, in order for the ventricle to receive blood, it has to be relaxed. But I wouldn't ask you about the specific details of the phases of the cardiac cycle. Here we have mentioned the resting membrane potential, the reversal of polarity, the depolarization, the repolarization, but there is one important thing that we have to know here is this plateau. In this plateau, what does this plateau mean? This plateau increases the duration of the action potential. First of all, what is an action potential? An action potential is a single wave of contraction. It is, why am I saying single wave? I'm gonna tell you now. It's a single wave of depolarization followed by repolarization. Why am I focusing on a single wave? Because if it starts to spread on the myocardium, then it is a nerve impulse. So what is a nerve impulse? It is a spread wave of depolarization or a spread action potential. But a single wave of depolarization and repolarization is an action potential. Why does it have a longer duration than the rest of the action potentials? For example, this is the action potential of an ordinary muscle, not of a cardiac muscle. This is to lengthen the refractory period, because you have here the absolute refractory period. What is an absolute refractory period? It is the period when the cardiac muscle cannot respond at all, however strong the stimulus is applied. The absolute refractory period includes depolarization and most of the repolarization. So when there is during repolarization this slowing down or this plateau, then you are lengthening the absolute refractory period. You are lengthening the period when the cardiac muscles cannot respond, and therefore you're giving more time to the ventricles to relax and to fill with blood. The electrocardiogram is a device or a medical tool that allows you to record the electrical activity of the heart. It's important because you can determine the heart rate, and therefore you can determine arrhythmias. You can determine whether there is ischemia or not, because unfortunately we didn't have time to cover this, but as I mentioned at the beginning, this is the ST segment in the ECG. And if you have angina pectoris, which is relative ischemia that we have mentioned at the beginning, then you will have depression of the ST segment. It would be under the zero line like this. And if you have myocardial infarction, which is more serious, you would have elevation of the ST segment. I wouldn't ask you about this, but it's interesting to know. And since we said that the ECG detects ischemias, then depression of the ST segment would be indicator of a relative deficiency of blood, whereas elevation is more serious because it indicates infarction and damage of the myocardium. Of course, you can detect chamber enlargement, and usually it is in the form of increased amplitude. So if the normal amplitude of the P wave is like this, if the atrium is enlarged, the amplitude would be increased. And of course, we can detect conduction defects. And the most important conduction defect that we can detect is the AV nodal delay. If there is a more delay than normal at the atrioventricular node, or a prolonged QT interval, which is very important. So these are the benefits of an ECG. And what are the normal ECG components? The P wave indicates spread of depolarization over the atria, so it indicates atrial depolarization. The QRS complex as a whole indicates ventricular depolarization, and it includes two negative deflections, Q and S, and in the middle you have a positive tall R wave. And then we have the T wave that indicates ventricular repolarization. And we said before several times that we can see atrial depolarization only, whereas we can see ventricular depolarization and ventricular repolarization. Why can't we see a wave for atrial repolarization? Because it is masked by the QRS complex. Why do we call it a complex? Because it consists of three waves. And then we have segments and intervals. An interval is more than a segment. An interval is a segment plus a wave. So for example, you have the PR segment that is made of the PR interval that is made of the P wave and the PR segment. And so the PR interval is the PR segment plus the P wave. So the PR interval represents what? Here at the beginning of the PR interval, you have to know where it begins and where it starts. Sorry, where it begins and where it ends. So here, for example, it begins at the beginning of the P wave and it ends at the beginning of the Q wave. So it represents the whole PR interval represents spread of depolarization over the atria and spread of depolarization across the AV node until we reach the beginning of ventricular depolarization. So it actually represents the duration or the interval of time between the beginning, you can define it in another way, the beginning of atrial depolarization to the beginning of ventricular depolarization. As for the PR segment, it just represents the transfer of electric impulses or the conduction of electric impulses across the atrioventricular node. The ST segment starts at the end of the S wave and ends at the beginning of ventricular repolarization, whereas the QT interval, which is more important, represents the both ventricular depolarization and ventricular repolarization. And here we have to mention that a prolonged QT interval, whether it is congenital or a side effect of medication, is very important to detect, because a prolonged QT interval might change into a fatal ventricular arrhythmia. Here you can see that the S1, which occurs at the beginning of systole, is just preceded by ventricular depolarization. And this is normal because depolarization and spread of electric impulses or generation of electric impulses on the ventricles should precede the contraction and should precede the closure of the atrioventricular valves. And then S2, which is the closure of the semi-lunar valves, occurs at the beginning of ventricular relaxation. And that's why we see it here after the ventricles repolarize and start their relaxation. So this is how you can adjust the heart sounds that you hear with the stethoscope together with the ECG. But this is an advanced piece of information that I would never ask about. Characteristics, of course, of the normal sinus rhythm are very important. It's a regular rhythm. It ranges between 60 to 90 beats per minute. The P waves are normal, meaning that they are upright and they are of normal duration. And very importantly, that one P wave precedes each QRS complex. The PR intervals are very important. Here we are talking about the interval between 0.12 and 0.20 seconds. The QRS complexes all look the same, and they have a narrow duration, which is 0.12 second, or a short duration, and the QT interval should never be more than 0.44 second. I will stop now because the video is now very lengthy, and I'll see whether I'll be sending another extension for the video to discuss the ECG cases and the mitral diseases. Thank you, and I wish you the best of luck with your final test.